Hello, everyone, and welcome. I am Maddie Dix. I am the alumni coordinator for the School of Nursing. We are so excited that you were able to join us for the second session of the School of Nursing's Hubs Research Series. As you all probably already know, the School of Nursing has an abundance of research programs that are aimed at advancing nursing science and practices. Within these programs, the school is home to 5 research areas, or hubs as they are familiarly known, which provide funding opportunities to foster collaborative interdisciplinary research programs. They feature faculty members and students with similar research interests, pursuing pilot projects on similar topics or themes, or integrating multiple existing projects. We are excited to continue this research presentation series with the Genomics of Patient Outcomes Hub. Dr. Yvette Conley is joining us today to represent this hub and present the amazing research happening within the School of Nursing that uses genomics and other state-of-the-art science omics approaches to advance the science of patient outcomes. Dr. Conley pre-recorded her presentation, which is lovely. So I will be sharing my screen for you all. However, she is here, as you can see her. She's live and ready to answer any and all questions that you may have after the presentation. So if you have any, please feel free to add them into the Q&A session, the chat function, and I will be facilitating those after the presentation is finished. So without further ado, I will share my screen, and I hope that this sometimes, it just starts to freak out, and I'm just like, okay, let's see. All right, Dr. Conley, I am looking at you. Can you see this, the movie?
I see
a black screen.
A black screen, yeah.
Perfect. Okay, that is exactly what I'm looking for. Okay. All right, so I'll go ahead and hit play.
Piece that sets our research hub apart from other
Is there
hubs
audio
on
now?
Pitt campus.
Okay. Yes.
The Patient Outcomes Hub also ensures that our next generation of nurse scientists are equipped with content and methodologic expertise that will ensure that nursing is represented as precision healthcare initiatives are implemented into nursing practice. So what type of omics approaches are folks in our members in our hub using? So omics is a broad term that's used to encompass any of the omics like genomics, transcriptomics, proteomics, metabolomics, microbiomics, epigenomics. And I'll talk a little bit about what each one of these are and what they can tell us. But basically the umbrella of omics means that you are using one of these technologies to advance science. So our hub members use genomics, which is where you would look at the variation in DNA sequence to better understand patient outcomes. You could use transcriptomics or messenger RNA levels. So whether a gene is expressed or not, and the extent of expression for a gene can have implications for some of the phenotypes that our hub members are interested in. Proteomics, where we're looking at protein levels. Metabolomics, where we're looking at metabolite levels. Microbiomics, where we're looking at the types and the quantity of microorganisms. And this could be virus, this could be bacteria, this could be fungi. But any microorganism, because they have unique genetic material, if we know what their unique genetic material looks like, we can detect it and we can quantify it in our patient samples. And then epigenomics. With epigenomics, you're looking at the DNA, but you're not looking at variation in sequence of the DNA. What you're looking at is the chemical modifications to the DNA molecules. And a lot of that modification that happens is in response to our environment. And as a result, we can combine a lot of environmental and personalized genetic variation studies and look at how the environment actually impacts our genetic material, actually impacts our health. Okay. So I did want to put a shout out for, why we want to look at genomics within the content of environments. And when we think about environments, we usually think about, okay, in this particular environment, this individual is more at risk for a particular phenotype. What do we mean by phenotype? Phenotype is, that condition or that trait that we're interested in. So it could be blood pressure, it could be cardiovascular health, it could be any kind of chronic condition. It could be, presence or severity of a particular symptom like cognitive decline, pain, depressive symptoms, anxiety. If you think about some of the symptoms that neuroscientists are very interested in, those are phenotypes. And then we think about environment. And in this situation, we're just looking at two different environments, environment 1, environment 2. And we're looking to see, hey, does someone's genetic material play a role in helping us to understand the impact of the environment? Now, what might the environment be? The environment might be the neighborhood that you live in. It might be the neighborhood that you grew up in. It might be the, whether or not you've had exposure to, if you've been a smoker, so you've had exposure to smoke. It could be whether or not you've had exposure to a particular intervention or therapy. And we know that if we look at two different environments and we look at a particular phenotype, that what someone's genetic material looks like might actually help us better understand why some people do better in some environments than other people. So when we look at this, we look at genetic variation and we see that, okay, across environment 1 and 2, maybe folks with certain genetic material might have a higher predisposition for a particular phenotype than another. Or maybe they have the exact same risk. But then we look at this and we see, okay, hey, environment 1 versus environment 2, everybody's risk goes up for that particular phenotype, but some people's risk goes up more, which is what we're seeing here in particular, where folks that have this genetic endowment in environment 2 has an increased risk over folks who have this genetic endowment. But what's really interesting and where really we want to be taking into account environment as well as omics is look at this environment 1 and environment 2, where we have folks with one particular genomic endowment, their risk going up, versus another group with a particular genetic endowment, their risk going down in the same environment. So as environments change and people move into different environments or they have different exposures, some people are going to be more at risk for the phenotype you're looking at. Some people might actually have reduced risk. And you can imagine how if you were measuring the phenotype and you were measuring the environments and you were not taking into account that omic variation, how you could completely miss the environmental piece. So people who are looking at environment and exposures need to look at omics, and people who are looking at omics need to look at environment. This is another exemplar to show you how omic variation, in particular here we're talking about genetic variation, but how it impacts different levels and different steps across the disease continuum. So we have behavior, we have personal exposure, we have absorbed dose to that exposure, we have early biologically effective exposures. We have alteration to structure and function that then results in early disease and then disease progression. There's lots of factors that are feeding into this disease outcome and predication for the disease outcome, things like age, things like diet, things like constitutional health and comorbidities, but we can't ignore the genetic factors. And then we bring in the epigenetic factors where you're looking at the exposures, these environmental exposures, and how those are impacting our genetic material, which then are impacting disease. And I will say that, a lot of these factors that folks have linked to disease outcome, like age, like diet, as an example, We know that age impacts epigenetics. We know that diet impacts epigenetics. We know that certain medications impact epigenetics. So again, when we think about that nursing lens and we think about what nurses are interested in from behavior to disease outcome, if you're not taking omics into account, you really do risk picking up on the behavioral component or picking up on the environmental component. So really what we should be thinking about is doing research that encompasses the biologic pieces with the behavioral pieces with the environment and looking at everything together. I wanted to also say that one of the things that with our hub that we are very interested in, and we've had many discussions, we have our hub has a weekly meeting. And in that weekly meeting, we have lots of discussion about current state of the science. We have lots of discussion about resources that we should all be thinking about using. It's really a once a week time where we can all come together and we can all discuss some of the new things that are on the horizon, or this person learned something and they want to share it with the group. Hey, I figured out, how to do this, or have you seen this resource? Let me walk you through it. And oh, hey, I went to this workshop and here's what I learned. So the object of those weekly meetings is to support one another, to also keep ourselves up to date, and to also be talking about how we should be using existing resources. One of the existing resources that we're all really enthusiastic about in our Omics Hub is the All of Us Research Program. And you can learn more about the All of Us Research Program at allofus.nih.gov. But this is the National Institutes of Health initiative. So this isn't, nurse specific, this is NIH specific. The National Institutes of Health is putting a lot of resources into the All of Us Research Program. So the All of Us Research Program, is really, what they're trying to do is they're trying to get at least 1 million people enrolled into this study. They are looking to enroll folks from diverse ancestries, diverse ethnicities, making sure that we have good distribution across a variety of population, that we really truly are representing the diversity of the US population. And they are looking to find folks who are willing to do several things. One, allow continual access to their electronic health records for research purposes, so that the All of Us program can analyze data in a de-identified manner that comes out of your electronic health records that could, if we had access to those records for 1 million people, what could we learn? The other thing, in addition to access to your health records, is to also have participants fill out specific research-related instruments and tools to collect data. And we'll talk a little bit about one of those tools that, one of those areas where tools are being utilized for research purposes through All of Us. The other thing is you offer up a biologic sample. So, and from that biologic sample there, the All of Us program is going to generate omics data on all of these folks who are enrolled into All of Us. The idea here is that we will be able to link omics data with the electronic health records, as well as the additional research instruments that you fill out as a participant. And what'll happen is they'll be able to look at, this person developed this phenotype, and this is what their genetics looks like. This person had this reaction to this intervention, and this is what their genetic material looks like. Hey, when we bring in ancestry, we see that folks from this ancestry seem to be more impacted by this particular intervention than another. So really what we're trying to get at is, instead of using this one-size-fits-all approach, which has been the approach to healthcare for a long time, what the All of Us program is trying to do is to generate the evidence to say, at some point, we would like to take someone's demographics, someone's clinical history, someone's family history, someone's omic variation, someone's, environmental exposures, take all of these things into account and be able to say, hey, we think this is what you're at risk for. Maybe we could mitigate, proactively some of that risk. But also, prior to having an intervention, who is more likely to benefit from this intervention? And if we have a menu of interventions, maybe there's a better way of figuring out who should get what intervention, because this is the intervention that we, based on evidence, we think is going to work best for you. And maybe for that person over there, not only would it not work for them, but they might end up having a lot of side effects and a lot of adverse reaction to a particular intervention. So there's just a lot that could come out of this precision health initiatives that are going on at the NIH. And one of the things that our hub is very interested in is making sure that our hub members, and making sure that we're building capacity in our future nurse scientists to be able to sit at the table and be able to take advantage of all of us resources, but also contribute to all of us and use that nursing lens, so that we're holistically looking at these folks when we think about precision healthcare. Now, one of the things that I wanted to say was, when we think about these specialized instruments that all of us are using, COVID-19 instruments were developed by all of us. And so the participants in all of us, not only have they said, here's my biologic sample, here's my access to my electronic health records, but I'm also going to fill out some research instruments for you longitudinally along the way. And one of the bolus of instruments that they have had participants fill out have been those around COVID-19. And now they have a cohort of approximately 350 participants that have filled out these COVID-19 instruments. You have access to their electronic health records in addition to these COVID-19 instruments. And what they're finding out is that they're putting this data together and putting it out there for researchers to use. So for example, they came up with the COVID-19 Participant Experience Survey, or the COPE survey. They have antibody testing data. They have electronic health record data in COVID-19. So for folks who developed, who were positive for COVID-19, longitudinally, what do these folks show up with in their health records, as well as, hey, is there anything that we could look at either in our omics data or in the health records to say, this individual looks different. And that's why they had these outcomes post-COVID-19 infection. And maybe we could better understand, hey, folks in this age group with this biomarker had this outcome 6 months after their COVID-19 exposure. That's what all of us is trying to get into. And that's what we're trying to get across with the Omics Hub here in the School of Nursing, is that we were trying to say, folks, let's gather. Let's use existing resources. Let's make sure that nursing is at the table with all of this, and that nurses are using these resources, and that the nursing lens is applied to all of this information that's coming out of all of us, so that we are holistically taking into account the participant and not just looking at their genetic material, or not just looking at their demographics, or not just looking at their clinical situation, but that we're holistically looking at these individuals. And that's the nursing lens through which we want to encourage our hub hub members to take advantage of all of us. So who are members of the Genomics of Patient Outcome Hub? We have a large cadre of individuals who come to our weekly meetings, and these individuals, we usually have somewhere between 17 to 25 individuals who attend these weekly meetings. Members of our hub include undergraduate nursing students, so folks who are getting their BSNs, and the majority of them are conducting projects for their honors college thesis, and they are interested in something omics and patient outcome related. PhD and nursing students who are conducting their dissertation projects using omics are members of our Genomics of Patient Outcomes Hub, postdoctoral scholars and faculty from Pitt Nursing, as well as other departments at Pitt. And we do have a few members of our hub who are from schools of nursing outside of Pitt, but their School of Nursing does not have the infrastructure, the omics hub-based infrastructure in their home school. So they participate with us. So some of the patient outcomes that our members are invested in investigating. So we have hub members who are looking at omics and how it can help us better explain variation and recovery after stroke, variation and recovery after traumatic brain injury, variation in symptom development and severity of symptoms within the context of many common conditions, including a lot of chronic conditions and comorbidity of chronic conditions. Some of our hub members are looking at variation in symptom development and severity within the context of treatment for common conditions. So for example, we have quite a few folks in our hub who are interested in treatment for cancer. And within the context of having cancer and within the context of being treated for cancer, how does omics help us better explain the variation and disparity in development of symptoms in these patients? Some of our hub members are looking at variation in the development of delirium in the ICU. Some of them are looking at how omics impacts variation in pregnancy outcomes, variation in outcomes of neonates in the NICU, variation in response to interventions, including non-pharmacologic interventions. Our hub members have, they are well funded and productive. So currently we have 9 faculty in our omics hub who are NIH funded. Most of them have R01 funding or ROO funding. Some of them have R21 or RO3 funding, and one of them has KO1 funding. Our hub also, we're very proud of our trainees as well, across the spectrum from undergrad, PhD students and postdoctoral scholars. So the School of Nursing has been very lucky to have two PhD students who have received F99 KOO awards from the National Cancer Institute. Both of those individuals have been hub members. 2 postdoctoral scholars in our hub currently have K99 funding from the NIH. And our PhD students who are doing omics-based research for their dissertation projects have received NIH funding, foundation funding, including American Cancer Society, International Society of Nurses and Genetics, American Nurses Foundation, Oncology Nursing Society, Robert Wood Johnson, and Jonas Philanthropies, as well as internal School of Nursing funding. Our undergrad BSN students have received the Chancellor's Undergraduate Research Fellowship Award, Sigma's Rising Star Award, as well as internal School of Nursing funding to fund their undergraduate thesis projects for the Honors College that have an omics component. And we've been very productive. Many presentations, our hub members are very good at disseminating their research through presentations and publications. Our last reporting period, which was July 2019 through July 2020, our hub members published 57 manuscripts in the literature. One of the one funding mechanism that we are particularly very proud of is the T32. So this is funding from NIH to Pitt Nursing to fund the career development and education of 3 PhD students per year and 2 postdoctoral scholars per year. And that training program is titled Targeted Research and Academic Training Program for Nurses in Genomics. And these folks are the trainees that are supported by the T32 are embedded into the Genomics of Patient Outcomes hub and are integrated into that infrastructure. However, we have lots of students and lots of trainees who are not supported by the T32 that are also benefiting from the infrastructure of the T32, including our Genomics hub. Our hub members are also representing Pitt Nursing very well. They're out there, they're active in the professional nursing community. Hub members have been very instrumental in the development of the Omics Nursing Science and Education Network, which is a multi-NIH institute endeavor. It was an endeavor through the National Institutes of Health, NINRs, so the National Institute of Nursing Research, as well as the National Cancer Institute and the National Human Genome Research Institute. So these three institutes came together to support the Omics Nursing Science and Education Network. And key to the development of this network and moving this network along was, members of our Genomics of Patient Outcomes Hub. And this ONSEN effort focuses on education and training, so helping trainees find mentors. This is particularly important when it comes to folks who want to do omics-based nursing research, but they may not have the infrastructure or the resources at their home institution. And so they can come here to find mentors. Common data elements when we think about moving the science forward, including omics science within the context of nursing research, common data elements, so everybody's looking at things the same way, so that we can combine data and analyze it together is very important to our effort. And then research collaborations. So what this part of ONSEN does is says, okay, there are folks who have recruited hundreds, maybe thousands of individuals into their research project, did immense phenotyping of these individuals, and also banked samples or omics data for the effort. And those data really need to be shared. Those resources really need to be shared. A lot of effort went into generating them. Let's put it out there. Let's say, here's the resources that our members of ONSEN have available so that we could increase collaborations amongst nurse scientists. So our hub has been instrumental to the success of ONSEN, and ONSEN is paying it forward for nurse scientists. Our hub members are very immersed in the International Society of Nurses and Genetics. Our hub was at Pitt Nursing sponsored the first regional conference for the International Society of Nurses and Genetics in 2019. We have a past ISONG president and a future elect ISONG president, members of our hub for ISONG. We also have members of our hub that contribute to the education mission, the research mission, and the practice mission of ISONG. And ISONG's most recent Congress was held back in November of 2020. And we are proud to say that Pitt, our Genomics of Patient Outcomes Hub, our Pitt representation at ISONG for this meeting far outpaced any other school of nursing that sent folks for this conference. So we are very proud of the work that our hub is assisting the International Society of Nurses and Genetics to accomplish. We are also very reliant and very grateful for the infrastructure in the School of Nursing that helps the Genomics of Patient Outcomes Hub be successful. One of the key infrastructure pieces for our hub is the laboratories, the wet labs that are on the 4th floor of Victoria Hall in the School of Nursing. And I will say that when I first came to the School of Nursing, we were the first school of nursing in the country to have a wet lab that was designated towards the type of research that our hub accomplishes. We are no longer alone. A lot of schools of nursing have wet labs that can do similar things to what we can do now, but we were certainly the first, and we are very reliant on the wet lab infrastructure that is available in the School of Nursing. And in this picture, we have an undergraduate BSN student, a PhD student, as well as the lab manager, who is full-time staff in the School of Nursing laboratory. So without the infrastructure of the wet labs, we would not be able to in-house accomplish a lot of the training and a lot of the research that we do for the Genomics of Patient Outcomes Hub. So what are some of the thoughts that we have as we're moving forward as a group? We want to continue to support each other in order to maintain and increase funding and productivity, including keeping our weekly omics enthusiasts meetings going. We also are interested in continuing to recruit and support our trainees for both the T32 as well as other ways of supporting and recruiting top-notch trainees into our hub. We also, are trying to come up with ways to support the hub in a way that it can start to sustain itself. And, our members are talking about some of the efforts there. And then we are also building cross-hub synergy. So the Genomics of Patient Outcomes Hub is only one of 5 hubs that is supported in the School of Nursing. And one of the things that we have started to do is to have some cross-hub synergy where, they're members of our hub are also members of some of the other hubs in the School of Nursing, but then also bringing together some of those folks to have some hey, we come together, we accomplish more. And so one of the examples of where we're at and a goal that we have for this upcoming year is that we have folks from our hub, the Genomics and Patient Outcomes Hub, as well as members of the Cancer Survivorship Hub at the School of Nursing. And we've been collaborating, we've been acquiring pilot funds, and we've been moving efforts forward to submit something that's called a PO1 to the National Cancer Institute. And a PO1 is sort of like a large grant mechanism that brings together a lot of people, has cores, and has multiple research projects embedded under the umbrella of that PO1. And the idea here is that if the School of Nursing were to attain this PO1 from the National Cancer Institute, it would certainly support the efforts that are being put forth by the Cancer Survivorship Hub, as well as the Genomics of Patient Outcome Hub, but also will benefit the School of Nursing and the infrastructure in the School of Nursing tremendously to have this PO1 funding available to faculty, particularly junior faculty, as well as trainees in our school. So we have a lot of enthusiasm for the future. Our like I said, our Genomics of Patient Outcomes Hub members are disseminating their research findings through presentations and publications, and to keep that momentum going and support each other is certainly key to what the hub wants to accomplish in this next year going forward. And then I will just leave you with the thought that the Genomics of Patient Outcomes Hub brings together people, brings together resources and infrastructure, and that content expertise, that methodologic expertise that people bring to the hub. This provides a combined effect that is much greater than the sum of all of us working separately. So the goal of the hub is to keep that momentum going, to keep that synergy going, and to make sure that the School of Nursing is benefiting from the combined effect rather than the sum of all of us working separately. And so thank you very much for your attention. And if you would like to reach out at any point to get more information about the hub, any of our training programs, any of our dissemination efforts, And or if you think today you don't think of something, but tomorrow or next week you think of something, please email me and reach out, and I would love to maintain contact with anyone who has an interest in our Genomics and Patient Outcomes Hub. If you're interested in becoming a member of our hub, I would love to hear from you. If you're interested in hearing and learning more about what our hub is up to, I would absolutely love it if you reached out and communicated further with us. So thank you.
Thank you, Dr. Conley, for that lovely presentation. I now know that I can blame all of my problems that I have on my parents for their genes. So thank you very much for that.
Oh, that's not true.
I'm just going to tell them that. I'll be like, Dr. Conley told me I can do this. So, so there are a few questions. If you are able to answering some of them, have a few moments to answer them. Would you be able to talk about a little bit about your current research, what you've been working on in the in your time with the School of Nursing, at least your current stuff that you've been working on?
Sure. So I feel like we have a lot going on. You know, we don't focus on one project. I kind of take my omics toolbox to multiple patient outcome related issues. Two of the projects that I think we, that we currently have going on that are interesting. One is the patient outcomes after stroke, particularly subarachnoid hemorrhage. And what we have done there is we have taken cerebral spinal fluid daily from individuals who have had a subarachnoid hemorrhage, have come to UPMC Presby as standard of care. They get that cerebral spinal fluid drained. And what we do is we take that cerebral spinal fluid and extract DNA from the CSF. That gives us an idea of what the genetic material looks like that is probably best representing the central nervous system after that hemorrhagic stroke. We look at the DNA and we say, okay, the folks whose DNA methylation or epigenetics looks like this over the days after that stroke look different when someone is leading up to a delayed cerebral ischemia versus someone who doesn't have a delayed cerebral ischemia. And delayed cerebral ischemia is one of those complications that happens within the first 2 weeks after hemorrhagic stroke that actually has a huge impact on outcomes, how well someone does, whether they even get to leave the hospital. So mortality is an issue, but morbidity, how well they recover, how well they do, has a lot to do with genes being turned on and turned off during that recovery period in the acute setting. And so what we have found is that there is particular sets of genes that are turned on and turned off after acute injury that tell us when we look at this orchestration of gene expression, we're seeing for those particular genes, this person is at risk for delayed cerebral ischemia. We're trying to replicate those findings, but the take-home message is we may have a way to say who's at risk for delayed cerebral ischemia and intervene so that we can prevent that ischemic event that then has a big impact on patient outcomes. And the other project that we're particularly passionate about right now in my research is we're collaborating with folks who are doing breast cancer research and folks who are enrolled into randomized controlled trials for exercise to mitigate the impact of cancer, breast cancer, and treatment for that breast cancer in cognitive decline. So we are looking at what genes are turned on and turned off, what genes are orchestrated in an individual within the context of breast cancer, treatment for breast cancer, and who is going to have that cognitive decline. Not every woman experiences it, but who's going to experience that? If we could figure out who's going to, we could mitigate that risk. Who benefits from the exercise intervention as well. So we're taking our omics toolbox to that patient population to better understand mechanistically what's going on when someone has cognitive decline within the context of treatment for breast cancer. What happens? Why does one person have cognitive decline mitigated by exercise while another person doesn't? We're trying to figure out mechanistically what's going on there. Figuring out that mechanistic component gives us a lot of ammunition to better patient outcomes overall. I don't know if that answers your question.
Yeah,
Molly,
no,
but,
absolutely,
yeah.
absolutely.
those are a few things that we're doing. Yeah.
Yeah, it's just, I mean, just a little snippet of what you're working on.
And that's me, but we've got a lot of members in the hub doing really cool stuff.
Oh my gosh, yes, I can imagine. Yeah. So of course, everyone's mind right now is COVID-19 and how that affects our daily lives. Are there any findings on the long-term effects of COVID? I know you talked about it a little bit with all of us and how the different people who have tested positive are able to do a little bit of research into the omics of it, but are there any long-term effects of it on like different populations, if you found any?
I mean, I think that's the question to be answered. I don't think we have that answer. Certainly all of us is setting us up to be able to answer those questions, to say, hey, here are the folks who tested positive, and here's all the sequelae. But we also know that some people who test positive, have very mild, if any, indication that they have been infected. So first of all, what's different? What's different about the individual who's infected and has a very mild or really no course, versus someone who escalates and has a lot of is impacted tremendously health-wise by that infection? Could be lots of things. We know about comorbid conditions, chronic conditions, age, all those things. But if everything taken into consideration controls for all those things, and we still have great variability, as I'm going to tell you right now, genetic variability that you bring to that infection, how you and your genetic material respond to that infection is going to explain some of the variability in who has severe sequelae and who has mild to no sequelae within the context of COVID-19. And I think the verdict is still out as to, physiologically, what happens within the context of a COVID-19 infection, and how is that impacting outcomes 6 months, 12 months, 18 months, 2 years from now. We know that there are people who were infected in the 1918 epidemic who had long-term effects from that.
Yeah, it's crazy. I mean, I feel like everything changes daily too with COVID, and how the difference, like it affects different people with, like one person will have a fever and then the other, like, and that's the person who tests positive won't have it. So it's like, why? Like, why is that happening? So that's definitely very interesting. Absolutely. There is a registrant on the call who is doing a research proposal on omics. So maybe they would be willing to reach out to you to talk a little bit more, but they had a question about the ontology of resilience and the stress of abuse and trauma. Is there any evidence of that?
Repeat that again?
Yeah, of course. Is there any evidence on the ontology of resilience and the stress of abuse and trauma?
All right. So this is a burgeoning area. So we know that early childhood adversity impacts it chemically modifies people's DNA, and that chemical modification impacts gene expression. And so we know that there are traumatic events. We know that there are adverse events that happen in someone's life where, it translates into pathophysiology. How does it do that? It probably does it. At least one mechanism is working through how that stamps our genetic material differently because of that adverse event. Now, when we think about recovery after something like brain injury, there's ongoing research that says that the type of injury that you have is obviously very important to that recovery. But beyond that, omics certainly plays a role in some of that variation. And we have some folks who in our colleagues of ours who are looking at early childhood adverse events, traumatic brain injury, and how all of that comes together to change our genetic material, and then that impacts the outcomes that we see. And this is predominantly being done in a pediatric population. So there's a lot of cool stuff going on. And so when you think about that omics proposal, and whoever that is, if you need help, reach out. But when you're thinking about that omics proposal, making sure that you're talking about, what's going on in the environment, could be that adverse event, could be, whatever that exposure, everybody wants to think of exposure as like, a chemical exposure, or, something that you're exposed to in the environment, fracking and chemicals or something like that. And that's true, but you gotta remember that some of the exposures are behavioral exposures and adverse exposures. And how that impacts our genetic material, but also, and then how that translates into health, or impacts on our health. But also, this person brought up resiliency, and we know that coping and resiliency, there are omics components to how we cope and how and as well as our resiliency. Yeah, there's literature out there on that.
Yeah, absolutely. All right. Well, I think that is about it. We're almost close to the hour. So I wanted to thank everyone once again for joining us on their lunch break, because it is lunchtime. If you have any questions, please feel free to reach out to me or to Dr. Conley. I will share, if Dr. Conley, if you are amenable to doing this, I will share your contact information in a follow-up email to all of our registrants, so they have
Absolutely.
your contact information. And if you want more information about any of the hubs, there is a ton of information on the School of Nursing website. And just a little pitch that the hubs is one of the featured funds for the School of Nursing's Pitt Day of Giving. So if you were at all inspired by Dr. Conley's presentation and you want to support the cause, please feel free to connect with me. We are hoping to continue on with our hubs presentation, so keep an eye out for an upcoming email for the upcoming research presentation. We would love to see you all again virtually. Thank you guys so much, and thank you, Dr. Conley, for your presentation.
My pleasure.
It was lovely.
Thank you, everyone.
Absolutely. Thank you guys so much. Have a wonderful rest of your Thursday.
Take care.
Bye.
Bye.